Hello everyone. In this video, we will show you how to do samples analysis for both solutions prepared or samples prepared under the pH solubility profile construction experiment and the effect of co-solvent ethanol on the solubility of mefenamic acid experiment. Remember to always clean thoroughly all equipment, tools, and working area before and after working. And also remember that many of these steps are and should be done parallel. First, remove your bottles from the shaker and place them in a well-organized working area. Start with rinsing the filtration assembly. Here we are using a gravity filtration. Then we need a funnel and Erlenmeyer flask together with a filter paper. Rinse everything using distilled water at the beginning. All Erlenmeyer flasks that will be used for filtration and also for samples dilution should be labeled clearly using section number and group number, as well as the solvent, buffer solution, agent, and pH. Always prepare a beaker labeled as waste to collect all remnants or residues. Fold the filter papers and place them in the funnel. Then do the wetting step using distilled water. Do the same for all of the 6 filtration assembly. 3 to 4 drops of distilled water are enough to wet the filter paper. Now discard the remnants from each filtration assembly, Erlenmeyer flask. Now start pouring a small amount from the tested solution in the glass bottles in the specified gravity filtration assembly as labeled. Here we have to discard the first portion of each filtrate. Why? This is due to the fact that this portion will have lower concentration than the real sample concentration, as part of the sample material in the sample will be absorbed on and absorbed in between the filter paper fibers. Do the same for in all experiments. Specifically this one, I cannot emphasize enough on the importance of labeling. Now, pour more volumes on each funnel, again as labeled. The poured amount or volume should be controlled carefully, such that you will You should never reach the side or the limit of the filter paper, since this will create problems, as the unfiltered solutions or particles may leak out from behind the filter paper, leading to turbid solutions. Turbid filtrates are problematic, whatever the reason to form them, as the filtrates will have undissolved solute, and this will lead upon dilution to overestimated solubilities higher than the real samples or solute solubility. Meanwhile, waiting for the filtration to complete, prepare the Erlenmeyer flasks, the small Erlenmeyer flasks that will be used for dilutions. Remember that we will make 1:1 dilution with 0.1 or 0.2 molar NaOH. This will be done by accurately measuring equal volumes. Again, volumetrically measured volumes equal volumes, which means that two volumetric pipettes of the same volume should be used. Here we are using 10 ml volumetric pipettes. One should be labeled as sample and the other one should be labeled as sodium hydroxide, NaOH. Clean these pipettes using distilled water. Place enough quantity of water in a separate beaker and use it to clean your pipettes thoroughly. In a clean labeled beaker, bring sufficient amount of 0.1 or 0.2 normal NaOH, depending on the available for you. While waiting for complete filtration of your samples, prepare yourself for the dilution by placing 10 ml of the used sodium hydroxide using the labeled volumetric pipettes you cleaned and prepared in each of the small Erlenmeyer flasks that will be used for the dilutions. Here the dilution factor will be 2. Dilution factor is equal to final volume divided by the initial sample volume. The final volume is the total volume taken from the diluent, sodium hydroxide, and the sample itself. Then in our case, this will be 20. And the initial volume is the volume that we will withdraw from our samples. And this is 10 ml. Then dilution factor here will be equal to 20 divided by 10, which is 2. After generating your filtrate, try to complete filtration up to the end. Withdraw using a clean 10 ml pipette volumetric pipette, 10 ml from each filtrate, and place it as labeled in the Erlenmeyer flask used for dilution. Always remember to rinse thoroughly the volumetric pipette using distilled water in between samples before and after withdrawing the filtrate. As we already know, the most accurate tools, volumetric pipettes and volumetric flasks, as well as analytical balances, should be used while preparing samples, diluting samples, preparing buffer solutions, and calibration curve construction. As you can see, the volumetric pipette we are using to withdraw samples are being washed, cleaned, rinsed using distilled water in between samples. Please maintain a very well organized, ordered working area. This will make things much easier for you, and you will be always in complete control on your work. You will be evaluated based on this. Why this dilution step? Why do we need to dilute our samples? You know that we will analyze our samples using UV visible spectrophotometric analysis, and using UV spectrophotometric analysis require the use of a blank. By definition, a blank is everything in the sample but the substance of interest, in our case mefenamic acid. If we tried to filtrate our samples, then immediately measure their absorbances, we will need to do the blanking step or the zeroing step each time for each sample, which means 6 times, because each sample will have a different blank since it's being prepared using different solvent. By performing this step and diluting our samples by 2-fold, we are converting our sample, actually half of our sample, to the same solvent, leading to the fact that we can use the used sodium hydroxide as our blank. And we will not need to do the zeroing steps 6 times. Actually, we will only do it once for all of the section. Only one time using sodium hydroxide. And here it is worth mentioning that we selected sodium hydroxide because our samples are being prepared at different pHs, and one of these pHs or two are higher than 7. By using sodium hydroxide, this will shift the pH of all samples toward a high pH that will not affect the solubility. After completing the dilution step for all samples, measure the UV absorbance at 287 nanometer, which is the lambda of measurement corresponding to our substance of interest, which is mefenamic acid. As you all already know that UV spectrophotometric analysis have some requirements. These include the lambda of measurement, which is in most of the time the lambda max of the substance of interest, as we mentioned, and the blank. Start with cleaning the cuvette. First, we always clean it with distilled water several times, then using the blank also several times. After doing so with our cuvette, fill it with our blank, which is again sodium hydroxide as we discussed earlier, and place it in its position in the correct direction such that the transparent surfaces or faces toward the UV spectrophotometer beam, light beam. Now, adjust the lambda by pressing on set nanometer button or go to wavelength button. It depends on your used instrument. Then, after doing so, zero your UV spectrophotometer via pressing the measure blank button or zero button. Now you are ready to measure the absorbances of your samples. Always start with solutions where you do expect to have the lower concentration of the substance of interest. Here we do expect the lowest solubility at the lowest pH. Then we will start measuring from citrate buffer pH 3 solution. Continue measuring the absorbances and recording of the results. Remember to rinse the cuvette in between measurements using distilled water. And it's necessary to completely dry the cuvette before placing it in its position inside the UV spectrophotometer. Using a very soft tissue. Do further dilutions using sodium hydroxide, the used molar concentration, whenever you need to do so. When do we need to do further dilutions? When we get absorbances higher than the linear range, which is in our case 1.2. Absorbance values higher than 1.2. Then, how to calculate the overall dilution factors? The overall dilution factor is simply the multiplication of dilution factor 1 and dilution factor 2. Then it will be equal again to dilution factor 1 times multiplied by dilution factor 2. Always dilute samples with absorbance values more than 1.2 with a suitable dilution factor. For example, start with the dilution factor equal 5. How? Withdraw 1 ml of the sample using a volumetric pipette and complete the volume with the used solvent up to 5 ml in a volumetric flask. Remember, this will be the second dilution factor. Repeat the same procedure for samples prepared to study the effect of ethanol on the solubility of mefenamic acid. Now, salt formation practical part 2. First, we need to confirm the sodium salt formation via melting point measurement. Remember that melting point is recorded as a range. Then, second, we will measure the aqueous solubility of both sodium mefenamate, the prepared salt, and mefenamic acid using approximate solubility concept. To measure the melting point, We should start with filling the capillary tube with both our salt and a second capillary tube with mefenamic acid. Melting point measurement will be done using the same procedure you used during your previous courses, including pharmaceutical organic chemistry course. The capillary tube should be first filled with not more than half centimeter of the tested powder. Remember to tap it to the closed end. Again, to confirm the formation of the salt, we have to measure the melting point of the prepared salt and the melting point of mefenamic acid, the raw material. What will confirm the formation of the salt is if we ended with two different melting points. Then confirmation by the generation of two different melting points ranges. Place your samples in the melting point apparatus and watch for melting. Do the same with mefenamic acid. Now watch for melting. After that, we will measure the aqueous solubility of the prepared salt, and we will compare it with the aqueous solubility we will measure for the acid, mefenamic acid. We will use again the approximate solubility concept. Again, we are measuring the solubility of mefenamic acid using the same method, approximate solubility, for comparison. To do so, measure 300 milligrams of the prepared salt, sodium mefenamate. Use a measuring cylinder to measure 3 ml of distilled water and add this volume to the 300 milligrams of sodium mefenamate. To test the aqueous solubility of mefenamic acid, please weigh 100 milligrams of the acid. Then place the measured quantity in a labeled beaker as we did with the salt. Measure 3 ml of distilled water using a measuring cylinder and add them to the Measure the quantity from mefenamic acid in the beaker. After shaking, mixing, you will find that sodium mefenamate is soluble in water, which means its solubility is at least 10%. Then the salt solubility is more than 10 gram percent, while you will find that the acid is insoluble. Consequently, its solubility is less than 3.33 gram percent. Special thanks go to Mr. Hamid Al-Tahir, the lab technician, and thank you all.